Ankhuri Singhal welcome you all to the world of RNA. Sidney Altman once referred to the RNA world as a hypothetical state in the origin of life on Earth. It existed even before the modern cells evolved. RNA stores genetic information as well as act as catalysts for autonomous replication even without the need of proteins. The first molecules to possess such properties must have been chemically simpler polymers but resembling RNA. Backing the concept of RNA world are viroids, which according to their discoverer are more plausible living relics of the pre-cellular RNA world than introns or any other RNAs. Viroids are the first representatives of subviral pathogen domain composed of single strand of circular RNA of just 246 to 401 nucleotides, lacking protein code as well as protein coding ability, suggesting their emergence even before ribosomes. They even possess ribozymes, which are considered as the signature of the RNA world. Thus, viroids are the oldest RNA replicons, which like RNA fold up to form secondary structures with conserved motifs, short structural elements and functional domains, which determine their mode of infection. Viroids are known to cause significant economic crop losses worldwide, exhibiting pathogenic diseases in different hosts. And one such host harboring a large number of viruses and viroids is grapevine, in which, due to vegetative propagation, exchange of propagation material globally, and perennial life cycle, associated pathogens spread and mix rapidly in different combinations, resulting in complex diseases, thus compromising the quality and productive life of grapevine. Viroids. India is known to stand at second position in the production of grapes in the world, with an export earning of rupees 1900 crores. Yet, the productivity of grapevine has steeply declined in the past few years, which may also be attributed to these virus and virus-like diseases. Grapevine is mainly known to be infected by 5 viroids worldwide: grapevine yellow speckled viroid 1, grapevine yellow speckled viroid 2, Australian grapevine viroid, citrus hexacortis viroid, and Hoxton viroid, of which HSVD and CVD have wide host range and they cause significant losses in other crops. But along with AGVD, they cause latent infection in grapevine, while GYSPD1 and GYSPD2 produce yellow speckled symptoms at higher temperatures and vein banding when co-infected with grapevine family virus. Due to these mixed infections, wide host range and frequent mutations, exclusion of viroids from elite germplasm is considered important, which is where a good diagnostic protocol becomes necessary. But there is a lack of information even on characterization, quantification of losses, and infection of these grapevine viroids in commercial grapevine genotypes of India, as well as the detection is also time consuming and costly, as conventional PCR requires 5 to 6 hours and a thermal cycle for diagnosis. Realizing this, I started my research endeavor on characterization of viroids infecting grapevine and development of a rapid diagnostic protocol with the following 2 objectives. The first objective was detection and molecular characterization of viroids infecting grapevine in India, for which grapevine genotypes were collected from Delhi and Maharashtra and leaf samples were used for total RNA isolation, which was subjected to 2-step RT-PCR assays followed by cloning in PGMT vector for sequencing, which was analyzed through BLAST. Published as well as designed primer sets were used for the detection of all viroids, and the primer set giving specific amplification was further used for detection in 23 grapevine cultivars. And Thompson seedless, Sharad seedless, and black prints were found to be co-infected by all 4 viroids, while Cyrax sessions, black musket, Bharat early, beauty seedless were found free of all 4 viroids and thus may serve as potential sources of viroid-free planting material. In the next step, the phylogeny of above viroids was studied to know their evolutionary relationship with isolates from other countries, and it was observed GYSPD1 Indian isolate clustered along with isolates of Pakistan, Iran, China and Thailand, while GYSPD2 Indian isolate clustered along with isolates from Croatia, Pakistan, South Korea, Iran and Italy. Hoxton viroid Indian isolate clustered along with isolates of Iran, Hungary and Croatia, while AGVD Indian isolate clustered along with isolates of Iran, thus suggesting their possible introduction from these Asian, European and South American countries along with planting material and vice versa. With a high sequence identity of 96 to 99% between the isolates, the secondary structure of GYSPD1 Indian isolate was found similar to the reference sequencing, with slight structural differences in terminal right regions. Similarly, in GYSPD2, Structural differences were observed only in terminal right regions, while in case of Australian grapevine viroid, in spite of high sequence identity, marked structural differences were observed in terminal left and the pathogenicity domains. But a characteristic central conserved region of genus Axca viroid could be located and found similar in all the above three viroids. While in case of Hobston viroid, characteristic central conserved region of genus Hostoviroid was located with structural differences in the variability domains. Thus, nucleotide substitutions, additions and deletions were observed in the sequences of Indian isolates from the reference sequence. The second objective of our study was to develop a rapid diagnostic protocol for which reverse transcriptase decombinase polymerase amplification was standardized, which outcomes the lacuna of all other existing detection techniques of viroids. For this, leaf samples were crushed and total RNA was isolated as well as crude sap was prepared and added to lyophilized reaction mixture, which was incubated in dry bulk, thus dispensing the use of thermal cycler, and was observed on gel, followed by cloning, sequencing and gene bank submission. Specific RPA primers were designed for each viroid, which are of 30 to 35 nucleotides in length, and they were first evaluated through RT-PCR, and the primer set giving specific amplification was further used for RT-RPA assays using RNA as well as crude sap as template, and was observed that the results of RT-RPA were comparable with the results of RT-PCR both in case of RNA and crude sap as template, and we found that the time required in case of RT-RPA using crude sap as template was just 60 to 90 minutes, while for RNA it was 90 to 120 minutes, as compared to conventional RT-PCR, which requires 5 to 6 hours for diagnosis. As well as cost per sample incurred was also reduced by more than 50% in case of crude sap as template as compared to conventional RT-PCR. Thus, the primers and protocol developed in the study could successfully detect the grapevine viroids using RNA as well as crude sap. In a nutshell, 4 viroids were identified to infect commercial grapevine cultivars in India, with their possible introduction from Asian, European and South American countries along with planting material and vice versa. Viroid-free genotypes could be identified in our study, and this was the first successful attempt of RT-RPA amplification of grapevine viroids. Since exclusion and eradication principles are the only successful management strategy for viroid, thus viroid-free genotypes identified could be used as successful sources of viroid-free planting material, while the protocol and primer standardized for RT-RPA can be successfully deployed for rapid detection of grapevine viroids even in resource-constrained laboratories as well as national and international quarantine. Further, infectious clones can be prepared to study the infectivity assay of these viroids. And since gel-based detection is not possible on field, the fluorescent probes can be prepared to combat the same. And for simultaneous detection of these viroids, multiplex RT-RPA can be standardized. Till now, 2 publications have come out from my study. First, one published as the first report in plant disease with a NAS rating of 9.17 and other accepted in Indian phytopathology with a NAS rating of 5.90. I have also presented my work in Professor M.J. Narasimhan Award Contest at the Division of Plant Pathology, as well as secured my position in top 3 in many competitions held, and also served as MSc class, second year class representative in PGSSU. At the end, I would like to quote a saying by Max Planck: "A scientist is happy, not in resting on his attainments, but in the steady acquisition of fresh knowledge." My heartfelt gratitude to my chairman, advisory committee, faculty division of plant pathology, PG School, ICARI. Thank you.
Yeah, nice work. Our honorable chairman is saying that don't take comments easily. Nice work, but yes, really speaking, we also did MSc with simple electrophoretic pattern on isoenzyme proteins and so on. But now you people do a very good piece of work, so I feel like appreciating. This phylogenetic relationship which you try to make, what was the basis on that?
With the isolates from all other countries we collected, one isolate from other countries, and we developed the phylogenetic tree, and we observed that our Indian isolate...
Phylogenetically mean what? Generally in molecular biology we use some marker and make dendrogram and showing the relatedness.
Through neighborhood joining method we used for phylogenetic analysis.
Okay. Secondly, this RT-PCR which you subsidized, most of the time, preferably, we should not be having a primer length more than 50 to 20 nucleotide. The moment you increase the primer longer, there is a possibility of this primer binding at non-specifically, and when you do the PCR, you get not desired band, but more than one band. I was wondering why your primer was 30 to 35. To me, it was a bit longer. What do you say?
Yes, sir. In RT-PCR, our primers were of 15 to 23 nucleotides, 20 nucleotides only. But when RT-RPA were performed, its standardized size is only 30 to 35 nucleotides, which is specified with Twist DX, which is the patent. Actually, what happens in case of RT-PCR, this TM we are calculating and we give denaturation. That is why specific binding is there in the complementary regions. But in case of RT-RPA, we just perform it at 37 to 42 degrees Celsius. So denaturation is we are not giving. So for complementary binding, its length is specified a bit longer than the PCR primers.
And my final question to you: the moment you started your presentation, you put RNA word, on the screen. It impressed me. I thought, "Patani RNA ki puri population aa rahi hai." Tell me one basic question: why RNA is actually while DNA is relatively resistant? Basic question. Why RNA is alkyl oil?
Because of this single strandedness, it doesn't have, because DNA is double stranded and it will bind that double helical nature will give it.
70% of the RNA is a double stranded. It's not a linear as you think on the blackboard or in teaching, but it is coiled at least as much as 70 to 75%. That's not the reason. Reasoning technically is something else. Anyway, thank you very much.
Very simple query. Normally it is said that it is easy to identify the viral diseases in the number of crops, but it is very difficult, or maybe in most of the cases not possible to control when it appears. Why it is?
So these thyroid and viral diseases, we know it's very difficult to control, and thus exclusion is the one of the main principles through which we can avoid. Thus, these viral-free genotypes, whatever we can identify, may serve as potential sources which we can deploy for as mother stocks for the production.
Sir, do you agree with Dr. Vyas's statement that it is easy to identify viral diseases?
But yeah, it is very difficult.
Do you
Yes,
agree?
sir. Yes, sir. Yes, sir.
Easy to identify viral diseases based on symptoms?
Not based on symptoms, but RT-PCR we have
That is
to.
what he said. So both
Okay.
identification based on symptoms and management both are.
Yes, sir. Based on symptoms, it is difficult because many times they cause latent infection also. So symptoms are not there, through which we know that they are present.
Yeah, Dr. Jain, it's very easy. Whatever we cannot identify, we say it is viral disease. Simple. Okay, beta. Thank you very much. Thank you. Mr. Herman. Not yet.
Check. Good afternoon, all the dignitaries present here. My name is Dipankar Burman from Division of Plant Physiology. I welcome you all to my presentation. As you all know, that global warming is one of the major problems which not only affect the animal system but also affect the plant system. It reduces the productivity and increases the food security risk at global level. When we see in Indian condition, we observe that 95% of the rice producing areas comes under heat susceptibility. And one of the best example we can see here in Andhra Pradesh, where there was 10% yield reduction in case of rice in 2018, and that was because of heat stress. So we have to increase our productivity by 30% at the end of 2030 in India, and in global level we have to increase the productivity by 0.9%. The question comes that how we will feed the need of the future under this climatic change scenario. For that we have to know the basic physiology, how heat stress works. So here you can see that, a temperature of 33.7 degrees centigrade cause spikeless sterility in case of rice, and 41 degrees centigrade for 4 hours cause a complete sterility to the rice spikelets. That means the reproductive stage is the most important stage where heat stress affect, and the reason behind this is pollen viability and stigma receptivity. Under heat stress, plant generates lots of reactive oxygen species, which cause oxidative damage to protein, lipid, membrane stability, and also reduce photosynthesis activity. But these sessile organisms are very smart enough to tackle the heat stress. They have different mechanisms where Ross homeostasis is the most important mechanism. This Ross homeostasis is controlled by non-enzymatic antioxidant, enzymatic antioxidant, and also by different transmission factors, which are directly regulated by hormonal regulation. But there is a major question that how this Ross homeostasis can be controlled effectively and efficiently. For that, we go through the literature and we observe that there is a molecule called melatonin, which is ubiquitously present in all the kingdoms from animal to plant system. In animal system, it mainly regulates growth and development, but the major role in animal system in different stress tolerance. How this controls the stress tolerance? This directly scavenger Ross and indirectly scavenger Ross, and also improve the heat shock factors. In case of plant, we observe that it has a role in growth and development. You see here by different hormonal balance. And there are few literature which say that it has a role in abiotic stress tolerance. But there is no literature on heat stress tolerance. So ultimately we are here with two major scientific questions, that how Ross homeostasis can be controlled effectively and efficiently. On the other hand, whether melatonin have role in heat stress tolerance. With that, we hypothesize that under heat stress condition, application of melatonin will improve antioxidant activity, and ultimately it will improve the yield in plant system. With that, we gave our research title to decipher the role of melatonin under heat stress in rice by understanding different physiological, biochemical basis of stress tolerance. For that, we chose two contrasting genotypes, that is Pusa Basmati 1121 as thermosensitive and NL44 as thermotolerant. We use temperature 33 degrees centigrade and 41 degrees centigrade as a heat stress. And we also use two chemicals, that is melatonin and haloperidol, which are pre-standardized for treatment. Now the question is, what is this haloperidol? This is actually inhibitor of melatonin biosynthesis gene, and this is in case of animal. But for the first time in the world, we use this in plant system. We gave the temperature treatment in control chamber with the increase of 8 degrees centigrade, and the relative humidity was maintained at 51%. Different biochemical and physiological parameters were analyzed to understand the stress. At first, we standardized the melatonin content through HPLC, and we get the standard curve from where we observed that endogenous level of melatonin was higher in NL44 comparing to Pusa Basmati 1121. The most important finding here was that the endogenous melatonin content increases with heat stress specifically to NL44 comparing to Pusa Basmati 1121. Then we thought that what is the effect of melatonin on endogenous content, and you can see here there was 6-fold increase in Pusa Basmati 1121. On the other hand, the treatment of expected inhibitor reduced the melatonin content. That means this is the inhibitor in plant system. And for that we also checked the expression of the biosynthesis gene, and we observed that under heat stress, biosynthesis gene expression also increases. To understand the endogenous melatonin's effect on different heat-related traits, we observed that melatonin increases the membrane stability index very significantly in Pusa Basmati 1121. But the question is here that how it increases the membrane stability. For that, we analyzed two parameters for oxidative stress, that is hydrogen peroxide content and lipid peroxidation, and we observed that there was reduction in both the conditions. That means melatonin reduced the oxidative stress. But again question comes that how will it reduce the oxidative stress? It reduces the oxidative stress by increasing the activity of different SOD, catalase activity and peroxidase activity. But most importantly, it improved the total antioxidant capacity by 41% in Pusa Basmati 1121. That means it has role in direct scavenging as well as indirect scavenging also. So here you can see that we came to the conclusion that melatonin improves the ROS homeostasis, where it also improves protein stability and membrane stability. But as a physiologist, there is a major question that how melatonin improves physiological processes. Of that, we analyze net photosynthetic rate, where we observe that melatonin improves net photosynthetic rate in both the genotypes, but most importantly in Kusama-Basmati 1121. And the reason behind that was that improvement in electron transport rate in photosystem 2. And also, on the other hand, there was improvement in total chlorophyll content, which ultimately increased the net photosynthesis rate. But that was in case of leaf. Now, coming to the reproductive stage, that how it affects the reproductive organ. For that, we also checked the endogenous melatonin level in spikelet, and we found that 4 times increase in endogenous melatonin level in spikelet. That means melatonin has some significant role in controlling this reproductive organ development as well as his stress, which was directly correlated with the pollen viability, where we observed that pollen viability was increased by 41% in Kusama-Basmati 1121, and this resulted in the improvement of spikelet fertility as well as grain yield. As agriculturists, we are more focused to the grain yield. So we observed that there was increase of 12% and 21% grain yield in both the genotypes. So from here we can conclude that this Kusama-Basmati actually have lower level of melatonin content under his stress condition. But exogenous application of melatonin improves this endogenous melatonin content, which help this susceptible genotype to improve his tolerance to his stress. And finally, we conclude that application of melatonin improves direct and indirect scavenging of rocks, which ultimately give higher yield through spikelet fertility and photosynthesis rate. And future perspective, we can see that screening of germplasm with higher melatonin content for breeding programs to improve the stress tolerance, and also different formulation of melatonin which can be used directly to the field. And identification of melatonin signaling pathway was also important. Our work has been published in 2 journals with NAS rating 6.23 and NAS rating 5.31, and one is under review with NAS rating 9.67. We are very happy to say that this is the first report on melatonin in India, and also this is the first report in the world where we identified that haloperidol has inhibition effect in plant system for melatonin biosynthesis. I also participated in different national and international conferences and also different extracurricular activities. And I was also in the position of General Secretary in Plant Physiology Student Association. With that, I want to acknowledge my chairman, advisory committee, division of plant physiology, IARI, my parents, friends, and Almighty for their moral and economic support. I want to thank you all for your precious time. I hope you enjoyed my presentation. Thank you.
Question, please. Mr. Burman,
Yes, sir.
a nice presentation and good work.
Thank you, sir.
But my question is out of the box. That question is ultimately we are working for the farmer of this country.
Yes, sir.
Can you deliberate this way to the farmers?
Yes, sir. Actually, what we observe here, melatonin improves stress tolerance, right? So, if we can formulate some dose of melatonin, it can be used by the farmers directly in the field when there is stress. And most importantly, I have not discussed here, this melatonin has only abiotic stress tolerance also and different other abiotic stress tolerance also. So that means in all the stress conditions, we can use melatonin as a spray. Because I also use as a spray. So farmers can also use as a spray, and also it's economically also it will be good because its rate is very less in the market price.
Yeah. What is the chemical nature of this melatonin?
Chemical nature.
Yeah.
Okay. This is also this is soluble in water.
No. What is it made up of? What is the building block structurally?
Okay. Structurally, this is.
Is it a carbohydrate protein or a glycoprotein or
This is actually this is actually a carbohydrate molecule. Carbohydrate molecule.
What is its physiological role in the plant? When there is no
yes, in plant
when there is no heat stress.
yes, in plant generally it helps in signaling purpose. When it balance different hormonal condition, means hormonal balance, it helps in growth and development. Recently, there was identification of this receptor also in plant system in 2018. So people are saying that it might be a new phytohormone. So this field is also getting more interest for us to.
You mentioned about increased rate of photosynthesis and high productivity and ROS related some enzyme.
Yes.
You also included like catalase, peroxidase, dehydrogenase, which is good.
Okay.
But photosynthesis high rate you try to relate with the electron transport chain. Why you didn't attempt Revesco? Generally depends whether we are talking about C3 or C4 plant, but Revesco is the enzyme in leaf material probably.
You are very right, sir. Actually, we observed that in drought stage there was also role in Rubisco stability also with the melatonin. So, but in our research program when we designed, there was actually less time, and we are doing the work, but because of time limit, I was not able to show this presentation here.
Thank you. Thank you.
Yeah. Nice piece of work, but I have a suggestion to make before you arrive to a definite conclusion about the role of the melatonin.
Yes.
It's a good idea to take a couple of heat sensitive varieties, a couple of heat tolerant varieties, and then only you can come to a definite conclusion whether it is really playing any role.
Yes.
Based on one variety, you cannot lead to a definite conclusion.
You are right, sir. In future work we will see this thing. Yes, this is mood changing hormone.
So did Ashok's basmati change the mood? Very sure.
It actually controls the circadian rhythm in animal system, but in plant system, till now the scientists were not able to prove this. So maybe some people in future will prove some role in circadian cycle in plant.
Majority decided that song is not. Very much.
Yeah, now seed cells and technology. Mr. Yadav.
A very pleasant good afternoon to chair and juries, designated scientists and my friend. As we all know that India has around 31 million hectare of under wheat cultivation, which is highest in the world and second highest in the global production. Out of which 67% is under irrigated area and 33% is under dryland and poorly irrigated area, having 16% contribution to the production and very meager productivity of 1.57 tons per hectare in comparison to the 3.8 tons per hectare in irrigated area. So friends, my topic of interest is this productivity enhancement in dryland agriculture. Now, the problem in dryland agriculture is mainly irritating rainfall and surface moisture depletion, which leads to the low field emergence under these conditions. Low field emergence under these conditions are mainly attributed to sowing depth, use of shorter molybdenum genotype, and using less vigorous seedling. Now, the field performance in these conditions can be enhanced by increasing the sowing depth, for utilization of subsurface moisture, utilizing genotypes having long molybdenum genotypes for enhanced field emergence, and using vigorous seedlings for uniform field emergence. Now, enhancement of the productivity of wheat so far is mainly attributed to the semi-dwarf wheat varieties having RST1 and RST2 genes, having advantage of semi-dwarf stature along with the disadvantage of short colloidal length, which reduce the field emergence under deep sown conditions and hence the lower yield under dryland conditions. Now, how this problem of short colloidal length can be imbibed? There comes an idea of alternative RST genes such as RST4, 5, and 8, which having sorting stature along with the advantage of longer colloidal length, which yields which have better field emergence under dry land condition and high yield under dry land conditions. Now, in light of our background, I would like to introduce you all my topic of research, effect of gibberellin acid responsive reduced height genes on seedling emergence, seedling traits in wheat. Now, the research gap in this area are, there is no conclusive studies on relation between colorectal length and seedling emergence under dry land conditions. There is urgent need to have effective selection criteria for early seedling for dry land agriculture, and there is need to identify genotypes possessing reduced height genes on aid in variety development for target environment. Now, to fulfill our research gap, my study was planned with the following objectives: to study the effect of depth of sowing on seedling emergence and correlation to the colorectal length in advanced lines of wheat, to study the effect of seed size and embryo size on early seedling vigor, and screening of material for gibberellin acid responsive reduced height genes with available molecular markers. Now, my study was started with 130 genotypes, out of which 60 genotypes were finally selected on the basis of their colorectal length, and they are further subdivided into 3 groups of short colorectal length genotype, medium colorectal length genotype, and long colorectal length genotypes. Now, these are my experimental material along with their pedigrees. Now, to meet up the above objectives, my experiment was conducted to investigate, is there any relation of depth of sowing and seedling emergence for different colorectal length genotype groups? Now, for the seedling vigor parameters, we need to investigate root length, shoot length, and seedling drive, which ultimately gives us seedling vigor index first and seedling vigor index second. And along with speed of germination, it determines the potential of genotype to cater the target environment. Now, speed of germination, root length and shoot length was found highest in the case of longer colorectal length genotype, and the similar results were found in the case of seedling vigor first and seedling vigor index second. Now, the results of above experiment were clearly evident in the case of seedling emergence percentage, when seeds were sown at 3 different sowing depths of 5 cm, 7.5 cm, and 10 cm sowing depth. Seed emergence was found at par when seeds were sown at 5 cm sowing depth, and there is steep decline in the seedling emergence when seeds are sown at deeper sown depth, particularly in the short and medium colorectal length genotypes, in comparison to the longer colorectal length genotypes, which were found highest emergence. For the study of root morphology, root samples were taken and scanned under root scanner using WinRISE software, and the maps were generated using ArcGIS method. Now, which gives the root morphology such as root surface area, number of forks per seedling, and root volume. For the root parameters, surface area and number of forks were found highest in longer colorectal length genotypes, and the similar results were found in the case of root volume. Now, the above experiment concludes that longer colorectal length genotypes are more vigorous and had high emergence at deeper sowing depths. They also had high number of forks per seedling, more surface area, and root volume, and ultimately these genotypes have high efficiency to utilization of subsurface moisture and can perform better under dry land conditions. Now, for experiment 2, my aim was, can we identify an effective selection criteria for early seedling wearing wheat for dry land areas? For embryo oxygen measurement, wheat seeds were soaked overnight and it was excised and measured under ocular microscope. The embryo size was found highest in the case of longer colorectal length genotypes, and there were no significant difference found in the all 3,000 seed weights of all 3 groups. Now, this experiment concludes that long colorectal length genotype had longer embryo size, but there was no significant difference in the seed size of the groups. Hence, embryo size is a better selection criteria for seedling vigor in wheat. Now, my experiment 3 was conducted to investigate, is there any positive genotype in the material under study for non-gibberellin responsive RST genes. For the identification of genes, 40 genotypes from medium and long colorectal length genotypes were taken, and for the DNA extraction, leaves were taken and crushed in the extraction buffer, and after centrifugation, DNA was identified, and after identification of DNA, it was amplified using PCR and ultimately using GEDOC system. Scoring of SSR markers were done. 5 genotypes were found RST8 positive among 40 medium and long colorectal length genotypes, RST8 genotypes were found on chromosome locus 2D and link marker was XGWM261. The find extensions which were found positive RCLY 1621, 1630, 1636, 1641 and 1661, which having colloidal length range between 7.4 to 8.36. Now, salient findings from my study was longocolloidal genotypes are have better seedling vigor indices, higher speed emission, and better seedling root characteristics. These genotypes are more competitive for utilizing subsurface moisture and can perform better under dryland conditions. Amplifiers were found to be a more effective selection criteria for varieties than the seed size, and 5 genotypes were found RST8 positive, and all these had long colloidal length. Now, leads from my study is Amplifier is the more effective selection criteria for early seedling vigor in dryland areas, and identified 5 genotypes which were positive for RST genes and hence can be utilized for development of varieties suitable for dryland agriculture. Now, my 3 papers were accepted in different journals, and one is published online and one is in press. Now, I have also given an oral presentation at an international conference on BCM 2019, and at last, I would like to acknowledge my chairman and members of my advisory committee, head and professor division of seed science and technology, division of genetics, PG school IARI, ICR for the research fellowship, without which this research cannot be done, and at last but not the least, parents, teachers and friends. At last, I would like to bind up my presentation with the saying, for every dryland there is a cloud of revival. Thank you.
Thank you. Question, please. Dr.
For the genotype which you have identified for the gene, after cloning you must have sequenced them.
What's that?
When you have gone for amplification and cloning, you must have sequenced. What was the size of the gene you were?
So 192 bases.
You must have sequenced them.
Yes, sir.
But that you perhaps did not like to present.
Did you really isolate it and sequence it?
Yes, sir.
Why you will talk about that? I thought you just take it, known gene and then talk about it. How did you isolate the gene?
Yes, sir.
RFT जो डॉक्टर जैन पूछ रहे हैं आपसे, how did you do the isolation and send it for sequencing? कहाँ किया sequence?
Sir, division of genetics.
Huh?
Division of genetics.
Can you give me a protocol? How did the gene were isolated?
Sir, I don't have remembered all protocols.
What is the practical utility of your work?
Sir, to utilize subsurface moisture, which in valley area there is deficiency of moisture, and due to high temperature there is evaporation from the evaporation of water from subsurface moisture, and for utilization of subsurface moisture, this type of genotypes are helpful for which have longer colloidal length. So after sowing the seeds at the deeper sowing depth, that seeds can utilize that reserve moisture which was present in the subsurface of soil.
Do you know what is source-sink relationship?
Yes, sir.
What is source-sink relationship?
What proportion of food is to be utilized by source and what proportion of food is to be utilized by sink is a source-sink relationship.
Okay, thank you.
Thank you. Sink, just like wheat grain is a sink. Yes.
तो भी 1 मिनट के लिए आओ, आई सस्पेंड द प्रोसीडिंग। जो नई जीन और आइसोलेशन का नहीं बताना चाहते हैं तो भी ये बायोटेक्नोलॉजिस्ट पूछते हैं उससे जमाना कितना बदला है। I didn't see the gene and did the breeding and I was successful. I saw DNA only once when Dr. V.L. Chopra isolated. बाद में DNA नहीं देखा मैंने। आजकल इस कंडीशन में काम करने वाले प्रैक्टिकल काम किया है। अरे बेटे आप बताओ ना कि you have told selection criteria.
Yes, sir.
Of course I don't agree with that. I don't support that because once you have सिलेक्शन ऑन द बेस ऑफ एमडीओ डीजे प्रोसीडिंग का हिस्सा नहीं,
Sure.
तो उसमें ऑयल ज़्यादा होगा, चपाती क्वालिटी विल बी बेटर डेफिनेटली. नंबर 2 ये वेरी स्ट्रॉन्ग स्टेटमेंट शुरू में, नो कंक्लूसिव स्टडी ऑन इंडिपेंडेंस.
Yes, sir.
One of my teacher said once, never give absolute statement for logical senses.
Okay.
Now I tell you about this statement. Green revolution is not only liberty. Depth of soil was changed. Then first time ground root initiation irrigation. वो agronomic factors वो और भी होंगे. Then machinery had this pressure not come, we could not have been able to bring meat produce to home or to the market. That never gave such a, which is constantly proved earlier. Though they may not have studied all these things.
Actually, sir, I want to relate with this my research. That's why I'm.
नहीं, absolute statement ना दो, no conclusive ऐसा कुछ आना नहीं था.
Okay, sir.
क्या? नहीं पता है.
Yes.
Okay, thank you.
Thank you, sir.
Next is where am I? I'm lost. Anshuman Das from Soil Science and Agriculture Institute.
Honorable Chairman, respected jury members, distinguished scientists, and my dear friends, a very good afternoon to one and all. I am Anshuman Das from Division of Soil Science and Agricultural Chemistry, presenting my MSc thesis before you. Are we all not apprehensive about the constant increase of population in the world? Yes, we are. And we need to feed every hungry stomach adequately. But how do we do this? Well, we go for intensive agriculture practices with the help of modern machineries, fertilizers, pesticides, and with huge amount of labor and capital in order to produce more and more. But every coin has two sides to it. The other side of the coin reveals something which is not very appealing. Exhaustive removal of nutrients without adequate supply resulted in decline in most of the essential nutrients. And here I shall put my concern for potassium. 60% of yield depends on proper crop nutrition, which is why you need to do everything you can do latest about the soil fertility. So today's topic, the importance of potassium. In terms of amount of nutrients plants need to grow, potassium is only second to nitrogen. It supports water uptake, helps the leaf tissues to keep moisturizing instead of sucked up by the heat. It helps in better stalk strength and standability. The roots of the plants also need potassium to resist disease, drought, and stress. Potassium is also essential to maintain the different enzymatic activities as well as to increase the water use efficiency of the crop. But farmers of India do not apply fertilizer due to two reasons. Basically, they consider soil have adequate amount of potassium, and secondly, high cost of potassic fertilizer detach them from applying it. Do you know there is not a single kg of potassic fertilizer is produced in India? Yes, you heard it right. The total amount is being imported from European countries, resulting in high foreign exchange. But when the uptake of potassium exceeds supply, there is depletion in the soil solution potassium. As a result, in order to maintain it, more potassium gets released from the interlayer spaces. But this availability of interlayer spaces depend on types of clay minerals. International Plant Nutrition Institute also acknowledged the role of clay minerals in supplying the potassium to the plant. Do you think this is just a concept? Think again, this is the near future of Indian scenario. In order to address this, my thesis topic was potassium supplying capacity in soil under different land use systems of Assam. Under such background, my objectives were to assess the different pools of potassium in soil under different land use systems of Assam, to study the kinetics and quantity intensity relationship of potassium in soils in relation to mineralogy under different land use systems. So the soils were collected under 4 different land use systems of Assam, namely mulberry, forest, rice mustard, and rice fallow under 3 different depths in the order insectisol. To study the clay mineralogy, organic matter free soil was taken in a beaker and treated with CBD and kept in water bath for 80 degrees Celsius. After that, it is transferred to tube and centrifuged immediately. After centrifugation, it is passed to 300 mSieve to separate the coarse sand, and after 16 hours, top 20 cm solution is siphoned out to separate the clay from sill plus clay to study X-ray diffractometer. In X study, there is one side there is X-ray source and in another side there is X-ray detector. When an incident on a crystal, it is diffracted in a particular direction. The crystal that is act as a diffracting agent, and as a result, it is diffracted according to Bragg's law. The diskinetics of potassium was studied using 0.01 molar oxalic acid under 14 different time intervals, and potassium concentration in the supernatant was measured using flame photometer. Result indicated that pH of all the soils studied were acidic in reaction, whereas the cation exchange capacity in the rice fallow soil in 15 to 30 and 30 to 60 centimeter depth were highest due to presence of high amount of clay. To fulfill my first objective, different pools of potassium were studied. It was found that in the water-soluble clay, mulberry has showed highest amount with respect to other land systems. Whereas in case of exchangeable potassium, the mulberry has shown very much higher amount than the other land system. In case of non-exchangeable clay, mulberry also showed the highest amount, but unlike exchangeable clay, forest, rice mustard and rice fallow had showed higher amount. The total clay was comparable in all the land use systems of the Assam. The saline findings were averaged over the depth. All the soils had high to very high levels of non-exchangeable potassium. Exchangeable potassium was in medium range in mulberry soils, while it was in low range in the rice-based systems. In order to fulfill my second objective, X-ray diffractograms of clay samples were studied under 4 different treatments, and results indicated that elite peak was prominent in case of mulberry and forest soil with respect to rice-based system. Similar peak was observed in case of 15 to 30 centimeter as well as in case of 30 to 60 centimeter. Decomposition and profile fitting of the magnesium saturated glycerol solvated clay was done to know precisely and exactly the amount of nutrient. Similar decomposition and profile fitting was done for 15 to 30 centimeter as well as for 30 to 60 centimeter. Result indicated that the elite content was dominant in case of mulberry and forest in all the land system, which is indicated by red. Whereas in case of rice-based system, the kaolinite and interstratified minerals were dominant with lower amount of elite. Mica 001 by 002 was determined to know the relative proportion of dioctahedral and trioctahedral minerals. And it was found that mulberry and forest soil had shown the higher amount of trioctahedral mica with having value much more than 1 with respect to the other land use system, which indicates that the release of potassium will be more in mulberry and forest. Center of gravity was also determined to know the area weighted average of all the peak positions, and result indicated that rice fallow systems have the highest center of gravity value, which indicates that it has higher expanding type QC mineral which can be linked with the lower content of elite, and as a result less will be the release of potassium from rice fallow system. Cumulative release of potassium was studied, and it was found that mulberry soils had sent the highest amount of cumulative release due to presence of more amount of elite in all the 3 depths, followed by forest, rice mustard and rice fallow. Data obtained were fitted with different equations to know the release rate of potassium, and it was found that power function has shown the best fit with highest R square value. The initial release rate of potassium obtained from the power function was found to be highest in mulberry, which is in accordance with presence of trioctahedral mica, whereas the apparent release coefficient was found to be lowest in mulberry. Potassium fixation capacity in the rice fallow system was found to be highest in the 15 to 30 and 30 to 60 centimeter due to presence of more amount of clay. The quantity intensity relationship was studied to know whether the soil can supply potassium immediately or it can supply in long run. Result indicated that the slopes were steeper in case of mulberry and forest land use system, which indicates that the release of potassium will be more in mulberry and forest with respect to the rice-based system. Similar trends were observed in case of 15 to 30 centimeter as well as in case of 30 to 60 centimeter. Result indicated that the equilibrium activity ratio was found to be highest in mulberry, which is due to presence of high amount of exchangeable clay, which indicates that immediate fertilization is not required in mulberry with respect to other land use systems. Whereas the potential buffering capacity of potassium was found to be highest in rice fallow soils in 15 to 30 as well as 30 to 60 centimeter due to presence of high amount of clay, which indicates that immediate fertilization is required in rice fallow systems. The specific health was also found to be highest in mulberry due to presence of high amount of elite content with respect to other land use system, whereas the non-specifically held potassium was also found to be highest in mulberry due to having higher equilibrium activity ratio with respect to the other land use systems. The free energy of exchange was also calculated, and it was found that the rice fallow system had the higher negative value, which indicates that potassium has higher affinity to the solid phase, and as a result potassium concentration will be deficient in the soil solution of rice fallow system. Different correlations were studied between parameters, and it was found that elite content showed positive and significant correlation with the cumulative potassium release in all the 3 depths. And also the potassium fixation and potential buffering capacity also showed positive and significant correlation with clay in all the 3 depths. The second findings were the elite and elite-rich 2:1 interstratified mineral together in the clay fraction were noticeably higher in soils under mulberry and forest system than the rice-based system. The Mica 001 by 002 was positively correlated, while center of gravity was negatively correlated with cumulative potassium released from soils. Cumulative potassium released and intensity of solution potassium over time in soils under mulberry were very high compared to the rice-based system. So the conclusions are, soil clays with higher Mica 001-002 ratio and lower center of gravity would have greater potential for higher potassium release over a prolonged period of time. Both short-term and long-term plant availability of potassium were high in mulberry and comparatively very poor in rice-based system. The mulberry might sustain itself for a considerable period of time even with the potassium application, but rice-based system would require frequent application of adequate amount of potassium for sustenance. The soils under mulberry were most fertile with respect to extractable and non-extractable potassium and least fertile under rice-based systems. So the path is to introduce clay mineralogy as one of the important parameters for optimum potassium recombination and judicious management of potassium. Under my research, I have two research papers were accepted in the Indian Journal of Agricultural Sciences with 3 class of 6.23. Another is under review in soil research in class rating of 9.82. I have also awarded the Best Poster Presentation Award by Indian Society of Soil Science in 2017. I have also presented poster in International Conference on Climate Change, Biodiversity, Sustainable Agriculture. I have presented oral presentation in Annual Convention of Clay Mineral Society of India. I have attended National Conference on Remote Sensing by ISRO. One abstract for poster is also accepted by the forthcoming Agricultural Science Congress. I have also received first prize in Western Dance competition in Akas organized by PUC in 2016. At last, I have to acknowledge my contribution of my chairman, co-chairperson, advisory committee, division of science and agricultural chemistry, ICR JRF PG School, my parents, and division of agricultural physics. At last, I have to conclude with the words of John H. Baker. Clay mineralogy is a missing link in the potassium fertility of soil. Thank you.
Thank you.
Mr. Anshuman Das, I am interested to know what made you to select land use systems of Assam for this particular study?
Sir, actually the livelihood of Assam, livelihood of people of Assam mainly dependent on this 4 land use system. As Assam is the 3rd most highest silk producing state in the country, so from mulberry silk is produced. So I wanted to know that how this clay mineralogy is affecting the main land system under which the leaves of the people is under stake if these are not in normal condition. So for this I have collected this major land use system of Assam.
What are the factors in which influences the potassium supplying capacity of the soils?
The potential supplying capacity is controlled by quantity intensity relationship, release kinetics, different mineralogy, clay mineralogy, texture, clay content.
Can you little bit explain?
Yes, sir. In case of quantity intensity relationship, we saw that if it is found to be having more steeper, then the release will be more in that soils. Similarly, if in case of clay mineral, if we are having smectite clay, then the release will be less. Whereas if we have elite, then release will be more. So similarly, if we are having clay content, so depending on the clay amount, clay content and environment surrounding it, it will also depend the release amount.
Which type of clay has maximum amount of potassium?
Maximum amount of potassium is present in mica.
What is the physiological role of potassium in the crop plants?
Sir, actually potassium is a number of, it maintains activation of number of enzymes like tryptophan pyruvate, which plays important role in carbohydrate metabolism of plant. And also it is an important component in case of tryptophan.
Extract the cofactor for some of the enzymes activity.
Yes, sir.
Okay, thank you.
Thank you very much.
Thank you, Dasa. Next is Ms. Anusha.
Honorable Chairman of the session, esteemed judges, esteemed scientists, and my dear friends, a very good evening to all. Our diet comprises of vegetables, fruits, food grains, and oilseeds. Fruits and vegetables are popularly known as protective foods due to their richness in vitamins, minerals, proteins, fatty acids, and carbohydrates. Among different kinds of vegetables, eggplant is one of the Indian originated crops. In the whole world, China, India, and Egypt contribute lion's share in area and production. In India, West Bengal, Maharashtra, and Bihar are in the forefront. Among total vegetable production, 21% of production credit goes to eggplant. There is an amazing diversity with respect to fruit shape, size, color, and weight, and it is considered as the king of vegetables in our country. If we move on to nutritional scenario, solvent along with anthocyanin, phenols, and dietary fibers, and it yields nearly 35 kilocalories of energy. Thus, there are n number of evidences to boost this complement, such as anti-diabetic and anti-inflammatory action. So, due to this reason, brinjal is recommended for the management of type 2 diabetes by American National Diabetes Education. Although India is second in area and production, but we are still lacking behind in productivity, which is merely at 8th position. Many constraints came to the way, namely low yielding varieties, less exploitation of genomic resources, biotic stresses and abiotic stresses. Hence, these constraints leads to different lacunae, such as the yield level of hybrids and open pollinated varieties remains static, and QTL identification is limited in eggplant as compared to other solanaceous crops. So, in order to evacuate these gaps, we undertook an experiment entitled Genetic and Molecular Characterization of Fruit Traits in Eggplant. With this background, I am Banshri N from Division of Vegetable Science. Now, I am going to put forward my research objectives. Firstly, to elucidate the inheritance of fruit traits and anthocyanin pigmentation in eggplant. And the next is to validate the markers into QTLs governing fruit weight and anthocyanin pigmentation in eggplant. My first objective was to elucidate the inheritance of fruit traits and anthocyanin pigmentation in eggplant. In our objective 1, we followed biparental crossing, which involved Pusa Safed Began 1 of small and white fruited type, and another parent was Pusa Uttam of large and dark purple fruited type. The resulting F1 was intermediate between the parents, and F1 was self to produce total 168 F2 plants, which exhibited large variability for fruit weight and anthocyanin content. Observations like plant height, fruit length, fruit diameter, and fruit weight were recorded from parents and all 168 F2 plants. If we come to the results, the F2 population exhibited large variability for the traits under consideration. Among these traits, the variability for fruit weight ranged from 15.22 to 48.3 grams. We came across higher PCV values as compared to GCV, and heritability was more than 60% for all the traits. The above traits followed normal distribution, and skewness value was less than 1. Hence, our F2 population was most favorable for mapping study. Among different colors of brinjal, dark purple is highly preferred by the consumers. So, the F2 population exhibited variability for color ranging from white to dark purple. The fruits harvested at edible maturity were categorized into 9 different classes by using RHS color chart. The results showed that all the F1 were light purple in color, and in the F2, total 125 plants were having purple fruits, 31 plants were having green fruits, and 12 plants were having white fruits, which clearly segregated into the ratio 1:3:1, suggesting dominant epistasis. My second objective was to validate the markers linked to QTLs governing fruit weight and anthocyanin pigmentation in eggplant. To accomplish my second objective, freshly harvested leaves were macerated in liquid nitrogen and DNA was extracted by CTAR method. A total 241 SSR markers along with the reported QTLs were used for parental polymorphism survey. One second analysis was carried out by pooling the DNA of extreme plants of small fruits weighing 15 to 30 gram and large fruits weighing 200 to 248 gram. However, 3 polymorphic markers were identified between the parents and the bulks. Coming to the results, out of 241 SSR markers, 18 were found to be polymorphic between the parents, and these were used for identifying. My research